Okay, morning everybody. My name is Miss Moorhead and I'm here today to give you a taster session on BTEC Applied Science. So this is for any student who's interested in either the BTEC diploma, which as you know is equivalent to 2 A-levels, or the BTEC extended certificate, which is equivalent to 1 A-level. All right, so the purpose of today isn't really so much an information session, all right? It's to just give you an idea of what you can expect when it comes to doing the BTEC course. So I've got a very brief course overview for you. You can see here I've put the list of all of the units that you will be doing on the BTEC course. So if you're interested in the extended certificate, you would only do these 4 units that are in blue. Okay, and if you are interested in the diploma equivalent to 2 A-levels, then you would do all 8 of these units listed here. So just to give you a little bit of information, so the Principles and Applications of Science 1 and 2 cover all 3 aspects of science, that's biology, chemistry and physics, and those units are examined. All right, and then you've got science investigation skills, where you would be planning, carrying out investigations, analysing the data, writing conclusions and evaluations, and that unit is also examined. Then you can see the next 3 are more investigative, so there's lots of practical work involved in these units, okay? And then the last 2 units you can see are biology units with a focus on human body systems and how they work in good health, but also the effect of disease on their operation. Okay, so these 5 units here are assessed internally. Okay, so moving on then, we're going to do a little taster session today on cells, all right? So the idea of the taster session is we're going to recap a little bit of your GCSE knowledge of cells, and also then look at some of the applications for why we study cells, what are the benefits of studying cells. And then we're going to look at and identify some animal cell organelles that are both familiar to you, but also some new ones as well. Okay, so to start with then, I thought it'd be nice to have a little look at what you can remember from GCSE. So we're going to do a little Kahoot. So if you would like to log on to Kahoot on your device, okay, and I will put the Kahoot screen up for you. All right, so hopefully now it's just loading the game pin. All right, so if you would like to log on to Kahoot, you should see when you might want to do it on a different device. If you do it on your device that you're using, you should be able to see the questions as well. If you can't log on to Kahoot for any reason, then don't worry, you can still play along right by looking at the screen at home or school, wherever you are. Okay, so the Kahoot pin is 6569990. Okay, and you can just use the spin the wheel to give yourself a nickname. Okay, I can see I've got 1 player. Oh, I've got 2 players. Just going to give you a couple of minutes to log on. Not a couple of minutes actually, because we are quite short of time. So I'll just wait for maybe a couple more people. And then, as I said, don't worry if you haven't been able to log on at home, because you will still be able to see the screen and play away. Okay, so I am going to start now, so don't worry if you haven't managed to log on. Oh, I can see I've got more and more people joining. All right, so apologies if you haven't managed to log on, but you can still play along at home by answering the questions. Okay, well done everybody that got that right. So the subcellular structure only found in plant cells is, of course, the vacuole. Okay, so the next one then. Respiration takes place in this organelle. So is it the cell membrane, the mitochondria, the ribosome, or the nucleus? Just waiting for one more person to answer. Okay, well done everybody. Let's have a look at the answer. Oh, excellent. All of you that said it was the mitochondria. Right, some people have said the ribosomes. Remember, the ribosomes are where proteins are made. Well done, whoever the friendly llama is. Name the part of the light microscope that the microscope slide is placed on. Is it the tray, the platform, the plate, or the stage? Just love background music for this. Just waiting for one more. Well done, everybody. Okay, you got that right. It is, of course, the stage. Right, so we use microscopes a lot to help us understand cell structure. How do we calculate the total magnification? Slide size times objective lens mag, eyepiece lens mag times size of object, eyepiece lens times objective lens mag, or the number on the objective lens. Oh, well done, those of you that got that right. So we multiply the two lens magnifications together on the microscope. So, well, friendly llama, epic squid, and lively griffin coming up the leaderboard. Electron microscopes form an image of a specimen using light and glass lenses, electrons and glass lenses, electrons and magnets, or light and magnets. Okay, fantastic. All right, so electrons and magnets, those of you that got that right. So a bit more difficult. You won't have done much about the operation of an electron microscope. All right, you'll do more about that when you look at cells at A level. So light microscopes have a much higher magnification and resolution than electron microscopes. Is that true or is it false? Okay, well done. Those of you that got that right. So electron microscopes have a higher resolution and magnification. Okay, so we've got some new people coming up the leaderboard there. Well done. Magnification is calculated using the formula. Is it lens size divided by image size, lens size divided by real size divided by image size, or image size divided by real size? Fabulous. Those of you that have learnt your magnification formula, well done. Epic squid coming up the leaderboard there. Most animal cells retain the ability to differentiate even after they have specialized. Is that true or is it false? It is in fact false. Plant cells do, but most animal cells don't. Okay, that's why we can't regrow a new limb, for instance, if we are unfortunate enough to lose a limb. The image shows a specialized cell. What type of cell is it? Is it a sperm cell, a muscle cell, a leaf cell, or is it a root hair cell? Okay, it is in fact a root hair cell. Well done, those of you that got that right. Okay. And the next question, where in the cell are chromosomes found? Is it the nucleus, the Golgi body, the cytoplasm, or the mitochondria? is in fact the nucleus. Well done, those of you that got that right. Which is the correct order of size from smallest to largest? Is it gene, cell, chromosome, nucleus, chromosome, gene, nucleus, cell, cell, nucleus, chromosome, gene, or gene, chromosome, nucleus, cell? It is cell, nucleus, chromosome, gene. Ooh, Silver McCall in the lead there. In the image, which arrow is pointing to a chromosome? Is it X, Y, or Z? Okay, it is in fact Y. Well done. Actually, everybody got that one right. Okay, and the next one, the stem cell is a cell that has not yet changed to become specialized for a particular job. Is that true or is it false? It is true. Well done, those of you that got that right. Okay, got some new people on the leaderboard again. The area where stem cells are found in plants, is it known as the merry Xmas, the merry stem, the merry brandybuck, or the merry leaf? It is, of course, the merry stem, merry Xmas. Okay, well done, helpful meerkat, moving up the leaderboard there. An example of a prokaryote is it an amoeba, an algae, a bacterium, or a fungi? Well done, those of you that got that right. So fungi, of course, do have a nucleus. Prokaryotes don't have a nucleus, and that's bacterium was the correct answer. Oh, we've got a new leader, Lively Griffin. Excellent. Last question. Which two statements are true of eukaryotic cells? They're complex and have a nucleus. They're much smaller than prokaryotes. They don't have a nucleus. All animal and plant cells are eukaryotic. Okay, so it's the red one and the green one. Well done, those of you that picked both of those out. Okay, so let's see who our winner is. In third place, we have the helpful meerkat with 11 out of 16. Well done. With 13 out of 16 is the silver macaw. And with 13 out of 16, but faster, is the lively griffin. So whoever you are, well done. That is fabulous. Okay, let me just close that. Just mute that. Just mute that. Okay, so I'm just going to go back to PowerPoint then. Okay, so well done, folks. Right, so what we're going to do now is we're going to look at GCSE level structure of cells, of an animal cell, and then look at more advanced levels. So if you look at the diagram here, this is a typical animal cell diagram from a GCSE revision website. So there are 5 structures that you should be able to identify from this. Okay, so I know you can't participate unfortunately, but if you just do it in your head, and I'll go through it and see how many you get right. So if we look at number 1, it's the structure surrounding the outside of the cell. So it is, of course, the cell membrane, which controls what enters and leaves the cell. 2 is pointing at the liquid that bathes all of the other parts of the cell, all of the other cells' subcellular structures or organelles. and that's the cytoplasm, which you'll have been told at GCSE, that's where all of the chemical reactions take place. Okay, so number 3 is the second largest organelle that you can see there on the diagram, right? And that is the mitochondrion, right? Or plural is mitochondria, which of course is the site of respiration, so where respiration takes place. Number 4, you wouldn't actually be able to see these with a light microscope, but you are required to know that they're in the cell at GCSE, and they are, of course, the ribosomes, which are where proteins are made. And then finally, this structure here, right, the largest organelle within the cell is, of course, the nucleus, where all of the chromosomes are found, okay, and that, of course, controls the cell. All right, so why do we study cells then? Okay, so a number of reasons. So understanding cell structure and function helps us to diagnose illnesses such as cancer, right? So cancer cells actually often appear abnormal under the microscope. So a trained technician can actually look at tissue samples and may be able to identify some cancers. It also helps us develop treatments for the cancer. One of the things that you'll look at is gram staining of cells, all right, and gram staining is a method that we use to actually identify different types of bacteria, and then from that we can identify which of those bacteria will be susceptible to penicillin, so we can treat diseases that are caused by bacteria. Also, understanding different cell structures helps us understand why some athletes are particularly good at endurance events, whereas others are particularly good at sprints. So we've got slow twitch and fast twitch muscle fibers, right? And some of those, the slow twitch ones are adapted for endurance, whereas the fast twitch are adapted for sprints. Obviously very topical at the moment, so actually understanding cells and how cells can become infected is very useful to understanding how viruses can make us ill, and also developing treatments against those viruses. And another disease that affects cells is sickle cell anemia. So again, understanding cell structure can help us understand this disease. You can see on the left-hand side, we've got an electron microscope photograph of some healthy red blood cells. And then on the right-hand side, we've actually got some red blood cells taken from somebody suffering from sickle cell disease. And these red blood cells, you can see, are an unusual shape. They're sickle-shaped, which is where the disease gets its name from. And they can actually cause blockages in blood capillaries, which can obviously cause problems. Okay, so moving on to A-level study then, if we look at this video, all right, then let me just see if I can share. I think if I get to it here, okay, I want you just to watch this video and see how it actually, it gives a CGI of life inside a cell, okay, and just have a look at this and reflect on the complexity within the cell. Okay, so hopefully from that, you got some idea of the complexity of cells and how the fact that the cells are dynamic, the organelles move around and interact with each other. All right, so we're going to look at that now in a little bit more detail. Okay, so this is a typical advanced level diagram of an animal cell organelle. Okay, so you should be able to identify some of the organelles that we've already talked about, some of those subcellular structures. Okay, so if you look at number 1, you can see it's pointing to the mitochondrion, so you can see it in a little bit more detail there. Okay, number 2 is pointing at the ribosomes. Okay, so the little dots here are the ribosomes. Number 3 is the cell membrane surrounding the cell. Okay, number 4 is the nucleus. All right, and then you can start to see some new organelles or parts of organelles. So the nucleus is surrounded by a nuclear envelope, which actually allows substances to move in and out of the nucleus. And then you have the darker structure within the nucleus is called the nucleolus. And that actually is where the ribosomes are put together. Okay, and then they leave the nucleus. Then we have the rough endoplasmic reticulum, which is a series of membranes, and the ribosomes are on them, right, on the surface there, and that's what gives them the rough appearance. And this is where proteins are actually made and then transported through this network. Okay, and the smooth endoplasmic reticulum, very similar in structure to the rough endoplasmic reticulum, it just doesn't have the ribosomes on it, and this is where lipids are made. So lipids, remember, is another word for fats and oils. Okay, and then we've got the Golgi apparatus, which is often likened to a stack of pita bread, right? And again, membrane-bound organelles, and this is where proteins are modified and packaged, ready to be released from the cell. So things like hormones would get packaged up and released from the cell. And then finally, we have the lysosomes, which actually contain enzymes, digestive enzymes that will break down any worn out organelles that your cells are finished with. Okay, so we're going to look now at some electron micrographs. So these are photographs taken by electron microscope of various animal cells. Okay, so if you look at this, all right, this is part of a liver cell. So this here shows the nucleus, right? You can see the mitochondria here, and then various organelles here, right? So you can see some of the rough endoplasmic reticulum here. Okay, so have a look at this. See if you can identify any of those organelles that I've talked about. Okay, the ones labeled with the red line are what we're looking at on this slide. And I've put a textbook diagram for you to help you and the keywords at the bottom. Okay. So this structure here is the nucleolus. Well done if you managed to identify this. And then here we've got the nucleus or the nuclear membrane. Okay, so this one. You are here. Okay, so this is a diagram. Okay, so you're actually looking at the mitochondrion. All right, you can see it's actually got lots of membranes within it. Okay, and this is where respiration takes place. Okay, so here we've got a stack of membranes. This is the textbook diagram. All right, and this shows the Golgi apparatus. Right, so it's quite difficult to start with to distinguish between the Golgi apparatus and the endoplasmic reticulum. Okay, so they tend to just look a little bit neater and more like a stack. Okay, but like as I said, a stack of pita bread. Okay, and then here we have got so you can see these membranes here and they're rough. They've got ribosomes on them. So that's the rough ER. And then you can see between the bits of the rough ER, okay, we've got the ribosomes. All right, and then finally, we have got here, lots of membranes here, look like, again, all right, lots of membranes, must be ER. It doesn't have the ribosomes on. So it is, in fact, the smooth ER, all of this here. Okay. All right, so hopefully that has given you some idea of why we study cells, but also the difference in detail that you'll go into between GCSE and A-level. Hopefully you've learned to identify some different organelles. I'd like to thank you all for coming. Unfortunately, I don't have any time for questions. If you do have any questions, please direct them to either of these two addresses here. Okay. Oh, that's my timer going off. Sorry, telling me I've run out of time. Thank you very much, everybody, and goodbye.